Hello, my name is Lougee, and I'm a graduate student in the Peterson Lab at the University of Pennsylvania. Today I will be giving you a brief rundown on how we have taken advantage of isoxazole photochemistry to generate a new motif for photocrosslinking. Isoxazoles are five-membered heterocycles containing a nitrogen-oxygen bond that are abundant in drug-like molecules both in the form of fragment libraries as well as FDA-approved compounds. There has been some previous literature precedent showing that the NO bond in particular is sensitive to photoactivation, resulting in the generation of several photoreactive species, such as nitrines or azarines. Furthermore, many motifs commonly employed as photocrosslinkers, such as diazarines, arylazides, and benzophenones, are either prohibitively expensive, difficult to synthesize, or overbulky and perturbative. So being so incorporable in medicinal chemistry and with favorable photophysics, we posited that the isoxazole could be used as a photocrosslinker in chemical biology applications. So in order to test this, a small library of isoxazole containing small molecules were synthesized based off a scaffold previously identified in our lab that has been shown to bind the protein alpha-synuclein with a nanomolar affinity. So alpha-synuclein is a small intrinsically disordered protein that has the propensity to aggregate into fibrils, and it's this characteristic that makes it heavily implicated in neurodegenerative disease. So our initial set of probe molecules also contained a propargyl ether, enabling further downstream functionalization via copper-catalyzed click chemistry. From here, we were able to show covalent cross-linking to alpha-synuclein via fluorescence STS page, as well as validate the site of labeling and amino acid reactivity profiles of isoxazoles using tandem MS. To show the isoxazole's utility in chemoproteomic platforms, we then pivoted to the compound sulfisoxazole, an isoxazole-containing FDA-approved antibiotic. This molecule has shown some promise in the literature as an anti-cancer agent via inhibition of exosome formation. In order to fully understand the interactome of sulfisoxazole, a derivative was synthesized in one step containing a propargyl ether. The cellular interactome was then studied via a SILAC competition assay in MDA MB231 breast cancer cells. Proteins involved in extracellular vesicle cycling were found to be substantially enriched via gene set enrichment analysis. And this subset was primarily composed of heterogeneous nuclear ribonucleoproteins and contained the RNA recognition motif as their most prominent domain feature. EF-HAN proteins, as well as thioredoxin domain-containing proteins, also showed good enrichment, and representative protein examples, calmodulin and thioredoxin, were further confirmed to undergo cross-linking in vitro with sulfisoxazole using fluorescence STS page. In conclusion, we've shown that isoxazoles are capable of performing adequately as a photocrosslinker in chemoproteomic applications, as evidenced by their usage in protein binding site analysis, as well as interactome profiling with applications in drug repurposing and off-target profile generation. Thank you for your time, and feel free to reach out if you have any more questions.